chemotherapy. So let's see what is chemotherapy. Chemo means chemical, therapy means treatments. So chemotherapy means use of chemical agent, either synthetic or natural, that have selective toxicity for the cell of infection causing microorganism or malignant tumor without causing the harms to the living cells or tissues of human or animals. It is a therapy that used to the treatment of the cancer. Usually we use the chemotherapy. So the disease killed by the microorganism. So let's see the classification of chemotherapy. In chemotherapy, classified into main is two types. One is antimicrobial agents, or AME we called, or drugs. So drugs means we come to know that it is in the type of medicines or tablets or injections. So it is an agent either synthetic or natural. Natural means inbuilt of immune system we will get the disease will be controlled. Synthetic means we use the drugs to intake. So that kills or inhibit the growth of microorganisms such as bacteria and fungi. It is used to treat. So anti-cancer drugs are the used to destroy the kill the shrunken or the slow long growth of the cancer spreading. It is unused. So let's see what is antimicrobial drugs. So it is an agent that which kills the microorganism. Example penicillin or cephalosporus is the plants we will use it to the treatment. So in the main highly we can see in the diagram penicillin and cephalamides. It is used to treat the cancer. So microstatic is a chemical compound agent which prevents the further growth or the multiplication of the microorganism. It will be control the growth of the microorganism and the multiplication of the microorganism. It is an cephalamide. Higher exporter of the cephalamide is the Ireland. It is the highest medicine producer, and the USA is a import the medicines for the cancer treatment. So next will be the antimicrobial drugs or agents. High we can see the in a microorganism, bacteria, fungi, protozoan, algae or virus. In this picture we can see it is an antimicrobial drugs or agent. To kill these diseases we will consume some the types of the medicine in our daily lives. Let's see antibiotics depend upon their range of effectiveness. First one is a narrow spectrum of the antibiotics. Let's see what is narrow spectrum of antibiotics means, which destroy the limited range of bacteria only whether there is a gram positive or gram negative only. Example penicillin. In narrow spectrum it destroys only the gram positive or gram negative only in any two. If disease is caused, the gram positive will be killed, either the gram positive will be killed. So next come to the penicillium is the best example for the narrow spectrum for the diseases. So next is the broad spectrum antibiotics. Broad spectrum antibiotics means it destroy the wide range of the microorganism. Example gram negative and as well as the gram positive will be the treated for the broad spectrum antibiotics. And antiparasitics is the anti-protozoan means example like malaria. Malaria is caused by the stagnant water by causing the mosquitoes, or it will be caused. And anti-malignant is the example helminthes means example for the roundworm. Roundworm in our digestive system, the roundworm will be the present by consuming of the food or water we will get the microorganism. So it will be killed by the albendazole. The tablet will be we will be consumed for the anti-helminthes disease caused by. Anti-parasite means the parasite means what we food we will be eat, it will be consumed by the microorganism. So due to this it will be causes some diseases like stomach pain or fever or flu. We will get the diseases caused by this. So example will be the for the anti-helminthes is the albendazole. Then antifungal. Antifungal is the best example for Canidia. The fungal food where we preserved in the food material. Example we will kept in the refrigerator. Some vegetables we will get like the white color or brown color. The powdery fungal form will be formed. It is caused by the antifungal diseases. And antiviral. Antiviral is virus or hepatitis virus. It is the virus. It will be caused. Next, antimicrobial drugs. Antimicrobial drugs, it is depending upon the antimicrobial action. They are the classified as the inhibition of the cell and synthesis. Example, which is the penicillium, an anti-agent that will be inhibit the DNA virus. Andre in DNA virus, it will be caused the diseases. And anti-agent that involves in the RNA synthesis, and RNA synthesis also it will be caused the diseases. The inhibit the protein synthesis. In the synthesis of the protein, it will be caused the destroyed by the antimicrobial drugs. The inhibition of the folic acid and the synthesis. The example will be the cephalamides will be the causes. Next is the antiviral chemotherapy. Chemotherapy means it is the intake of the medicine. So here there is a further step. One is an antiviral of the cell penetration and uncoating, and numerites inhibitor, an inhibitor of nucleic acid synthesis, and inhibitors of the reverse transcription inhibitors, and nucleoside reverse transcription inhibitor, and non-nucleoside reverse transcription inhibitor, and protozoan inhibitor, and nucleotide analogs. Let's see one by one what will be further. So selected antivirate example will be, if we get a flu or any cough, we will take the some medicine. It will be act like the antiviral agents. So it will be control that microorganism or foreign material what will be causes by the against to fight against, we will get some immune. Let's see what is inhibitors of the attachment. Example, inhibitor attachment to the cell receptor is a virus specific events. Some inhibitors can be stopped this attachment to the virus to the cell receptor. Antibody can be bind to the extracellular and viruses and prevent this attachment. However, all through therapy will be antibodies is useful in the prolapse. It has been minimize efficiency in the treatments. So second one is the inhibitors of cell penetration and the uncoating. So in uncoating, some inhibitors will be several days steps in the viral replication. It will be stopped replicating, including viral uncoating. There is extremely selective. For selective, some it is definitely selective for that. This is what we take constituent. It will be act on further that process only. It does not harm to the other cell. It against only the influenza. Where it will be causes influenza, it will be act in the further step only. So next state, neuramides inhibitor. So neuraminide inhibitor is a new antiviral that selective inhibitor that neuramides of the influence of the A and B viruses. That neuramides is a selective for that relative the viruses from the infected cell. Where it will be infected cell, it is caused. It will be act for that influenza. So treatment which either with the influence of the system, where it will be caused pain, it will be affected. So next, inhibitors of the nucleic acid synthesis. Inhibitors of nucleic acid synthesis. Some antiviral agents are get by inhibiting the synthesis of nucleic acid and presenting. Most of the antiviral agents are nucleosides. Along that they are active against the viruses specific nucleic acid and polymerase and tripeptides. So it has some less active against the analogs, but it will be host enzyme will not get the affected. So it will be affected on where there will be causing agents. So in that only it will be act that. So in diagram we can see how it will be worked. In salicylic what the medicine will be take, it will be acted like that, it will be engulf and it get the immune system and inhibit the energy and it will be act. So next, tumor virus. So tumor is we come to know that tumor is mostly we use the tumor for the cancerous only. So in that cancerous there will be two types. One is DNA tumor, and in tumor viruses means tumor virus is caused by a cancer in the host cell. So most of these viruses possess relatively simple genomes containing small number of virus of the genes. Tumor viruses are developed extremely to the gene to disturb the complex regulating to the cell infected. So example, the viruses and tumor virus, there is two type, DNA tumor viruses and RNA tumor viruses. DNA tumor viruses means the DNA will be genetic material in some further humans or animals. In the absence of DNA tumor, there will be presence of RNA tumor. In RNA tumor also it will be translated from the diseases caused. Let's see, DNA tumor viruses means DNA tumor viruses has a two life form. One is the passive cell, another one is the viral replication causes cells in the cell death. In the non permissions, it will be viruses. DNA is mostly integrated into different sites of the cell chromosome. In the chromosome only, the tumor cells will be the passes by the parents to the offsprings. Next, RNA tumor viruses. RNA tumor virus is the genetic which will be retinoviruses. In retinovirus, there is more than the 30 different causes diseases will be found. In such, there is an extra non-structural genes will be allowed in the formation of the cell. In tumor, there is a different organic mechanism. In the sum, there will be the causing the RNA viruses in the rapid tumor development. In rapidly, it will be causes the diseases. Next, for this all diseases, we will be controlled by some taking the viral vaccines. That is, a prevention of the viral diseases can be achieved by the use of some vaccines that include the activities of immunity or passive immunity. So it will be inactive or killed viral vaccine contain viruses which have lost their ability to replicate their in order to get definite response to contain more antigen. It will be live vaccines. So the killing of the viruses and get the against fight against the viruses, it will be taking by the vaccines. Example, main vaccines are available for important viral diseases like polio and hepatitis. By this we will get the control of the diseases. The polio is the main in all the countries facing that problem. In the children birth we will get, for that we will get the vaccine. And hepatitis also we will get by the injection syringes. We will take the hepatitis injection. So thank you.